Good afternoon. My name is Lexi, and today I will be presenting my K Fellowship summer research on how different concentrations of microplastics affect the early embryonic development of the sea urchin species Echinometra mathii. First, I would like to thank John Kay for providing me the opportunity to conduct my research project through the K Fellowship program. To start off, I would like to introduce the topic of microplastics. So, microplastics are plastic debris under 5 millimeters and are often found in personal care, packaging, and cleaning products. And they're continuing to increase in their global abundance, but are becoming increasingly abundant, especially in subtropical regions in the Pacific Ocean due to the Great Pacific Garbage Patch, which indicates that Hawaii faces greater risks on the potential threats caused by the increasing microplastics in the oceans. Microplastics come from a wide variety of polymers, but polyethylene is a specific polymer that constitutes for approximately 71% of the microplastics found in the Pacific Ocean. It's most commonly utilized in plastic bags, personal care, and containers. And due to its overwhelming abundance, this experiment uses polyethylene microplastics because it also represents the most common microplastic pollutant encountered in Hawaii's marine ecosystems. And overall, microplastics in aquatic environments can enter marine organisms and humans through the means of trophic transfer. And trophic transfer is basically the movement of nutrients or contaminants through a food chain and results in biomagnification or the increase in the concentration of substances as they move up a food chain. Additionally, microplastics can enter marine organisms and humans through other means, such as ingesting foods containing microplastics or absorption either through water from marine organisms or the inhalation of microplastics in air particles for humans. But although it's known that microplastics can enter a wide range of marine organisms and even humans, there's limited knowledge on how microplastics impact important species. Recent studies have determined that microplastic presence is in the digestive tracts and gonads of various sea urchin species. Specifically, they found that the polymers polypropylene and polyethylene are most common, and the detection of microplastics in the digestive tract suggests that microplastics are ingested by the urchins, and the detection in the gonads suggests that microplastics can then spread throughout the body. This is important because sea urchins are vital for maintaining marine ecosystem balance, meaning that they're a keystone species, and they are specifically important because they help to control algae levels and prevent overgrowth. The species Echinometra is the most common species found in tropical warm water environments, therefore meaning that they're also the most common in Hawaii. And this was my reasoning for studying the species Echinometra mathii because of its abundance and ecological importance to Hawaii's marine ecosystems. Additionally, sea urchin embryos are easy to obtain, fertilize, have rapid synchronous development, and are easy to observe even at earliest developmental stages, like from 2 to 4 cell, and therefore serve as an optimal model for studying effects on embryology. Therefore, using the Echinometra mathii species and sea urchin embryos, my research question aims to answer how do different concentrations of polyethylene microplastics affect the early embryonic stages of Echinometra mathii. To conduct this research, sea urchins were collected from Kulia'o Beach Park and brought to acclimate in the lab tank. Then, after 3 to 5 days, they were injected with a 0.5 molar potassium chloride solution to induce muscle contractions, which caused the urchin to release their gametes. And as you can see on the left graphic, the production of white gametes signifies a male and orange signifies a female. If they were a female, they were inverted for their eggs to fall into a cup of seawater and then were washed for excess material. And male sperm was collected via a micropipette and stored on ice, then diluted before use. And then the sperm dilution was micropipette into the washed eggs for fertilization. Then after fertilization, embryos were added and microplastic water were added to petri dishes, and a pilot trial was conducted with the concentrations of microplastics based on previous research done by Trufogi. And in their experiment, they measured concentrations of 0.5 1, 5, and 10 milligrams per liter. Yet in my pilot study, I found that there was no significant impact from these concentrations, so we increased them to 5, 10, and 20 milligrams per liter. Then in the experiment, there were three concentrations and a control in which embryos grew without the presence of microplastics and had three replicates for each. Then for 2 days, the embryos were observed under a microscope and the first 10 were counted and scored. To backtrack, the initial determination of fertilization was decided by the presence of the fertilization embryo envelope, which can be seen within 5 minutes post-fertilization. And then counting started at 2 hours post-fertilization when the embryo should have reached 8 to 16 cell. And then at 24 hours post-fertilization for the gastrula, but in which we counted for gastrula cells, primary mesenchyme cells, and shape. But in the statistical analysis, we classified a normal gastrula by the counts found in the presence of gastrula cells. Lastly, at 48 hours, the embryo should have reached a pluteus, in which we scored for arms, shape, and pigment cells. But in our statistical analysis, used the data counted for the presence of arms. Using the counts collected during the time periods previously mentioned, the statistical analysis was performed using the Kruskal-Wallis test because the data was not normally distributed and there is a small independent sample size. Before going into my results, I'd like to restate my research question, which was how do different concentrations of polyethylene microplastics affect the early embryonic stages of Echinometra mathii? And after conducting the Kruskal-Wallis test, statistical analysis revealed that there wasn't a significant difference between normal, dead, and delayed counts at any time point or concentration. And on the graph, the x-axis represents the 3 observation times, and the y-axis represents the counts of embryos either being normal, dead, or delayed. And these results show that concentration did not have a significant effect on these early embryonic stages. So to further understand this, I looked into previous studies, which found that microplastics that have undergone aging conditions have enhanced toxicity impacts and inhibits the effects on cell growth of microalgae. And assuming similar impacts for the sea urchin embryos, I seeded microplastics in seawater, meaning that I set the microplastics in the water for a period of time before conducting my research as a proxy for aging the microplastics. And through this approach, I left the microplastics for 1 week before conducting my next trial. And results from this trial showed normal development at the earliest stages of the embryos, but did show a significant difference in the number of dead embryos between 24 and 48 hours post-fertilization. So these outcomes suggest that exposing sea urchin embryos to microplastic seeded water may impact their development process. And also that microplastics may release chemicals that make urchins more susceptible to the microplastic exposure, which aligns with other past research. But further research should still be conducted on how various aging treatments of microplastic impact echinometra embryology and possibly study into later developmental stages. I'll also be conducting further research which branches off of sea urchin embryology and moves into studying the impacts of UV irradiated microplastics on a monocyte cell line to assess for the microplastics impacts on human immune systems. Through this experiment, there were limitations that included the egg quality, the inability to control the volume of eggs per female urchin, meaning that females produce limited eggs, and the inability to determine the gender, meaning you can't physically determine the gender for spawning. And lastly, the difficulty to continuously collect sea urchins because Echinometra are a rock-boring species, so it's kind of difficult to pry them off rocks. Yeah. And this process overall taught me that embryology studies can be difficult to conduct for multiple trials due to the inability to determine gender for spawning and controlling the amount of eggs per urchin. But regardless, any research project will face its obstacles, so it's important to choose one that you're passionate about in order to persevere. And lastly, to give some acknowledgements, I'd like to thank my mom and my brother for coming with me to do my urchin collections. Um, Mr. Hill for helping me with my statistical analysis, Mr. Kobayashi for teaching me how to collect urchins, and Mrs. Kobayashi for guiding me through the research process and teaching me a lot about sea urchin embryology. Julia Miza for sharing with me her past research on sea urchin embryology and my other K fellows. Thank you.